In this video, we'll be discussing transcription and translation. These are the two processes that go together to make proteins from DNA and RNA. So let's dive into what are these two words. Let's start with transcription. Transcription is the process in which a portion of the DNA molecule or a single gene is copied into a complementary strand of mRNA. Hold your horses. What's that mean? Okay, it is the process in which one little piece that codes for one little protein is copied into complementary. Complementary means it matches the DNA strand of mRNA. So let's dive into the process of transcription. The very first step of transcription is identical to replication. Helicase is going to bind to the DNA molecule, except the difference is here, it's not binding at the very end of the DNA and unzipping the whole thing. It's binding at a very specific start codon or a start sequence that says we're going to start making this protein. It unzips the DNA to the point that needs to be copied, and then everything zips back together. So step one is helicase, binding to DNA and causing DNA to unzip. Step two is RNA polymerase, binding to the DNA and assembling a complementary, matching A with U, C with G, strand of RNA to match the DNA. Now, a quick note here. In replication, we used DNA polymerase. In transcription, we're using RNA polymerase. They are slightly different molecules, so you can't just say polymerase when you're talking about replication or transcription. You have to specify RNA or DNA polymerase. Okay, so we've started assembling the complementary nucleotides, so the A's and the U's and the T's, or the C's and the G's. Remember, when you're making RNA, if you have an A, where you would normally put a T, you're going to put a U or a uracil. Adenine with uracil, cytosine with guanine. The third step is RNA polymerase is going to reach a stop point. It's going to be a sequence of letters that says stop. Then that new mRNA, which is just a little piece of the DNA, a little piece, one gene, heads to the cytoplasm. Remember, RNA is a single-stranded molecule that can leave the nucleus. So the mRNA strand peels away from the DNA and goes to the cytoplasm. The DNA strand then zips back together and goes back to normal life. A quick word to remember when we're talking about DNA and transcription and translation is the word codon. A codon is a group of 3 nucleotides. Now we refer to codons especially during transcription and translation, for a purpose that you'll see more in translation, but we always refer to DNA in sets of 3 letters. So, for example, AUG, that's one codon. Another codon is CGU or ACG. Any combination of any 3 letters that come in a row make up a codon. So let's move on and talk about what is translation. So translation is the process by which we interpret a genetic message and build the protein. So when you're translating a text, let's say I've given you something and I've said translate this into Spanish. You are taking one language, English, and translating it, changing it into another language. The same thing happens in translation of RNA into a protein. So in nucleic acids, we speak in the language of nucleotides, A, T, C, G, and U. That's the language of DNA and RNA. Proteins speak a different language. They speak in amino acids. So methionine, threonine, serine, all of these other words that we're going to see more in a second are the language of proteins. Amino acids are the language of proteins, and then nucleotides are the language of DNA and RNA. So we have to translate that genetic message, those nucleotides, into a protein, or amino acids, and we use the ribosome to do that. So the very first step of translation is that mRNA that just left the nucleus, that single gene, travels to a ribosome. Ribosomes are protein factories. It meets up with the start codon. The start codon is always and forever AUG. That's the very first codon on mRNA that means to make the protein. So step 2 is that tRNA, transfer RNA, has an anticodon on one end. So that means it's opposite of the codon that you have on mRNA. And it has an amino acid on the other end, building block of proteins on the other end. It pairs the complementary codon of mRNA. So if we had AUG on mRNA, and we would have UAC on tRNA. Okay? And that specific tRNA that has AUC on the bottom, on the anticodon, carries an amino acid on the other end. That amino acid is methionine. Okay? And so that methionine that's how the transfer RNA is going to start to build its protein sequence. So the third step of translation is when the amino acid from one end of the tRNA breaks away and attaches to the next amino acid. So we start to make a string of beads of amino acids. So they are joined together. The string that holds them together is called a peptide bond. And the final step of translation is when that tRNA leaves the ribosome, or the rRNA, and the next piece of tRNA comes to the ribosome. This keeps happening until we reach a stop codon, or a sequence of 3 letters that says stop. This is the end of protein synthesis. This is the end of making a protein. All right, so let's look at a picture of what this looks like. It may help us to understand it a little bit better. So here at the top of your screen, you will see a strand of mRNA. It's got a bunch of different sequences on it, a bunch of different codons. So the ones that are active in the ribosome are GAG, GUG, and AAA. So those are your codons. These little things that look like T's are transfer or tRNA. At one end, they have an anticodon. At the other end, they have an amino acid. And each of those amino acids is a different thing. So this codon on mRNA matches up to the anticodon on the tRNA, and that tRNA is carrying a very specific amino acid. There are 20 total amino acids, and the sequence of these bases determines what amino acid they carry. So all of these tRNAs are meeting with the mRNA at the ribosome. They are passing along their amino acid. Their amino acid is making a peptide bond to the one beside it. And then the tRNA leaves the ribosome. And the ribosome keeps continuing down the mRNA until it reaches a stopping point. So how do we determine what the amino acid is that goes with our mRNA? So we have a couple of ways of doing this. We can use this wheel, or we can use a chart that I'm going to show you in just a second. But the first thing I want to show you is how to use this wheel. So the wheel corresponds to the mRNA strand, the sequence of mRNA DNA, or the mRNA sequence. So if the mRNA is CGA, you're going to start in the middle. The first letter of the codon is a C, so you're going to be in this quadrant, down bottom right. And you're going to go to the second letter, which is a G, and then to the third letter, which is an A. That codes for the amino acid called arginine. That amino acid is going to be coded for by anything that is CGG, CGA, CGC, or CGU. Always remember, what you're reading off of the chart is mRNA. It is not the anticodon. mRNA is what you read off the chart. So if our start codon is always AUG, then our start amino acid is always methionine. Okay, what's the other way that we can determine the amino acid? We have this table. So the first letter of the codon is in the left. So if the first letter is, let's use our start codon. AUG is start. So we'll start on the left-hand side of the table, A. Then we'll go to the second codon, or the second position, and that would be U. So now we're looking in this box here. And then our third letter is G. So we're looking right here at AUG, and that stands for methionine. So let's say you have a sequence of mRNA that is UAG. Your first codon is U. So you're going to start on the very first row of your chart. Your second letter is A. So you're going to go to the third column of the chart. And your third letter is G. So you're going to go down and find where U and A intersects with G, and that gives you a stop codon. You're always going to have methionine at the start and stop at the end of a protein. That's just how the protein making process works. So these two charts will be super helpful in helping you to determine what the amino acids that your mRNA codes for. So just to recap, protein synthesis starts with helicase that unzips the DNA. RNA polymerase travels along and matches it up. And then translation happens. tRNA comes to the mRNA, connects amino acids with peptide bonds. So that's a new protein strand with all of our amino acids. So RNA polymerase making mRNA. Once it makes its mRNA, the tRNA brings it to the ribosome, and amino acids are linked together with peptide bonds one at a time as their anticodon matches up to the mRNA. And as a final result of all of that, you end up with a newly minted protein.